Hello, my dear students. Welcome to my today's online talk on one more session of Genetics and Biotechnology, paper 6 of 5th semester zoology, prescribed by Bangalore City University. And today we shall discuss about CIB method of detecting the mutations. Okay. Now, so you can see here the Drosophila, commonly called fruit flies, which are popularly called Cinderella of animal genetics, because lots and lots of experiments and work has gone on these flies. And now today we will be learning about one particular topic with reference to these flies. So you will have to learn about the role of X-rays in inducing mutations. This is session 22 of your LMS Karnataka, Bangalore City University. Then I have divided the contents of my talk like this: to know about CIB method of detecting mutations. And now, Dr. H.J. Muller, a geneticist at the University of Texas, he worked on the effects of radiations on Drosophila and won the Nobel Prize in 1946. For his work on the effects of radiation on the production of mutation, he was given this Nobel Prize. And Muller designed an experiment to detect the presence of lethal mutation, as lethal mutations are more frequent than visible mutations. This method was called CIB method. For this, he created a unique female Drosophila that had a normal X chromosome and a special X chromosome which had special features. Okay? And that specially designed female, number one, it had an inversion, because of which crossing over was not possible with the other X chromosome, and therefore C stands for crossing over suppressor. And then it carries a recessive lethal gene I. Okay? And then it had a gene for bar I, capital B, that acts like a marker. And if the female Drosophila had the special X chromosome, she could be identified as she had bar eyes. Okay? So this was a special female Drosophila, unique female, designed by Muller for his experiments. Okay? Then, this is what I have told. Now, my duty is to explain this cross. Carefully observe this plate. I'll keep on describing this. So, the unique female, what he designed, was called CIB female. And this CIB female was crossed to a male Drosophila which had been exposed to X-rays. Okay, we now have a special female, CIB female, CIB female, and then we have a male Drosophila which has been previously exposed to X-rays, and our aim is to know whether exposure to X-rays has brought about any mutations. This is to understand X-rays as mutagens. Okay. Then in the F1 generation, you can see the plate. It consisted of females and males in the ratio 2:1. And among the females, 50% were normal with red eyes. The other 50% had bar eyes, which means they carried CIB X chromosome. So all females in F1 generation which had bar eyes 50% carried the CIBX chromosome. The males had normal round eyes. 50% of the males who received the CIBX chromosome died because of the lethal gene I. Please note, all living males were normal with round eyes, and the males who received the CIBX chromosome died because of the lethal gene. And when the CIB females of F1 generation is crossed to a normal male, the F2 generation will consist of only females. There will be a total absence of males, please note. This is because 50% of the males will die due to CIBX chromosome and the other 50% will die if the X chromosome that came from the grandfather who was exposed to X-rays had a lethal mutation. Please understand, I repeat, when CIB females of F1 generation is crossed to a normal male, the F2 generation will comprise of only females. There would be a total absence of males. This is because 50% of the males will die due to the CIBX chromosome and the other 50% of the males will also die if at all if they have received the X chromosome that came from the grandfather who was exposed to X-rays and therefore had a lethal mutation. Alright. So in this plate, this is what is explained. So F2 generation has no males, there are only females, and 50% of the females have normal eyes and 50% of them have bar eyes, which means that they have CIBX chromosome. So the absence of males in the F2 generation confirms the fact that mutation would have occurred in the male of the parental generation when it was exposed to X-rays. If mutation wouldn't have occurred in the male of the parental generation when subjected to X-rays, then at least 50% of the males with round eyes would have survived. And it hasn't happened. So the CIB female of the F generation has two abnormal chromosomes. One is the CIB chromosome and the other is the X chromosome from the X-rayed male, which has the lethal gene as a result of mutation due to exposure to X-rays. Even though she has two lethal genes, she survives. Unlike her sons who would die because the lethal genes are not alleles. Okay. This is what exactly the CIB technique tells you. So it is basically a method of detection of mutation. Dear students, this is a 10-mark question for your exams. If you write this cross pattern correctly, that would fetch you more than 60%. This is what all about the technique is. Now let us see some assessment questions. Before that I would have listed the references. So assessment question. CIB method for detecting mutations was proposed by Muller, Galton, Morgan, Gerard, and the answer is A, that is Muller. Assessment question 2. CIB method involves which of the following markers? Crossover suppressor, recessive lethal, bar eye, all the above. Invariably answer is D. Then assessment question 3. CIB method is suitable for scoring of sex-linked dominant lethal, autosomal recessive lethal, sex-linked recessive lethal, autosomal dominant lethal. Answer is C, that is sex-linked recessive lethal. So my dear students, I have taught you about this CIB technique. in detection of lethal genes. Okay, basically it was meant for studying the role of X-rays as mutagens. Thank you for your patient listening. Thank you. Stay blessed.